Look closely at a brain cell firing electrical impulses, and then look at a liver cell filtering blood. They look completely different, and they perform completely different jobs. Yet inside their nuclei, they contain the exact same sequence of DNA. If they are reading from the identical genetic blueprint, how does one cell know to become a neuron while the other knows to become a liver cell? Look at this classic double helix model. For decades, the traditional scientific dogma held that this static structure was your absolute destiny. We were taught that your DNA sequence was a rigid set of instructions, dictating everything from your physical traits to your maximum lifespan. But that rigid view has a glaring flaw. If you track identical twins, two people born with the exact same genetic code, as they age, one might develop asthma or heart disease, while the other remains completely healthy. Their genetic sequence didn't change. Instead, there is a hidden layer of biological programming sitting right on top of the genetic code, actively dictating how that blueprint is read by the body. That hidden layer is called epigenetics. The word literally translates to above genetics, referring to chemical modifications that change gene expression without altering the underlying sequence of DNA letters. Think of your DNA as the permanent hardware of a computer. The epigenome is the dynamic software running on top of it, constantly telling that hardware exactly what to do, when to do it, and how much to do. This software is highly sensitive to external input, acting as a responsive interface between your surroundings and your internal code. The macroscopic choices you make every single day, what you eat, how much you sleep, and whether you exercise, are translated into microscopic chemical tags that physically attach to your DNA. Your daily environment provides the direct signals that determine which of those inherited predispositions are expressed and which remain silent. To see how this works, let's look at a person pushing through a high-intensity interval training workout. This places severe temporary physical stress on the body. As muscles burn, a biochemical chain reaction fires through the tissue, crossing the cell membrane and plunging directly into the cellular nucleus. Inside, we find the DNA strand, but it is not bare. It is covered in a dense, complex layer of chemical tags. This diagram shows DNA methylation. Methyl groups attach to the DNA strand, acting as physical off switches, preventing cellular machinery from reading genes. Exercising drops the enzymes maintaining these blockades. With the methyl groups removed, the reader molecule glides across the strand. Previously silenced genes are now active, regulating energy metabolism, building new blood vessels, and repairing cellular damage. The mechanical stress of lifting a weight or sprinting up a hill literally clears biological blockades to activate dormant, health-promoting genes. This illustrates histone modification. To fit inside a cell, DNA wraps around protein spools called histones. When signals cause spools to clamp tightly, the DNA is hidden and genes silenced. When different signals cause spools to spread apart, the DNA is exposed, activating those genes. But this system responds to negative inputs just as efficiently. Scientists call the total sum of your environmental impacts the exposome, which includes chronic psychological stress, heavy air pollution, and nutrient-poor diets. A toxic environment adds harmful epigenetic tags. These instruct histones to spool up tightly, silencing the tumor suppressor genes that protect you from cancer, while simultaneously exposing the genes that trigger chronic low-grade inflammation. This is a core reality of modern medicine. Your inherited genetic predispositions load the gun, but your environment and your lifestyle pull the trigger. Unlike a permanent genetic mutation, epigenetic tags have a distinct characteristic. They are entirely reversible. By consistently adopting nutrient-dense dietary patterns, regulating your sleep, and maintaining structured exercise, you become the active programmer of your own biological software, scrubbing harmful tags off your DNA over time. This raises one of the most heavily debated questions in biology today. If our environment alters our epigenome, can we pass those acquired tags and the trauma that caused them down to our children? The phenomenon is called transgenerational epigenetic inheritance. It is well documented in plants and nematode worms and observed in mice. But in humans, proving it is incredibly difficult because of our long lifespans and complex cultural inheritance. Take a look at this photograph from the Netherlands in 1944. During the Dutch hunger winter, the population suffered extreme famine. Decades later, researchers found altered DNA methylation patterns and higher rates of metabolic disease in the descendants of the women who survived that starvation. It is cases like this that keep scientists investigating how far epigenetic memory can reach. Regardless of how the generational debate settles, the immediate impact of epigenetics on your own body right now is an undeniable scientific fact. Human biology is highly plastic. Every meal you eat, every walk you take, and every full night of sleep you get is an active molecular signal, one that optimizes your long-term health and actively defends your body against chronic disease.